Hello, my name is Ben Westphalen, and it's a great pleasure to be part of this year's OncoAlert Colloquium. And I would like to showcase my favorites, so to say, in the field of tumor agnostic therapeutics. These are my conflicts of interest, none of which have influenced today's talk. Over the last couple of years, we have seen, spearheaded in the US, but also taken up in Europe, a lot of tumor agnostic approvals, namely in MSI cancers, NTRK fusion positive cancers, in tumor mutational burden high tumors, and then RAD fusion and HER2 overexpressing cancers, last but not least. So the concept of tumor agnostics is now well established, and in the following presentation, I would like to discuss a couple of upcoming tumor agnostic targets and ways to therapeutically address them. This is what I would like to discuss today: NRG1 fusions, MET amplifications, MTAP loss, targeting HER2, and then targeting RAS with a focus on innovative strategies. So starting off with NRG1 fusions, a common theme is that fusion events, targetable oncogenic fusions, are exceptionally rare. You can see ranging from 0.1% to sometimes around 0.5%. And what you can see on this slide is that there are various fusion partners. And what happens if you have an oncogenic NRG1 fusion, you have an EGF-like ligand, and this leads to HER3 and HER2 activation, which then leads to proliferation and survival pathways. Colleagues from the University of Heidelberg were able to show that using broad HER targeting agents such as afatinib was effective in NRG1 fusion positive pancreatic cancer. This was published by my colleague Christoph Heining in Cancer Discovery in 2018. Now we have a specific drug called Zenocutuzumab, which is a bispecific that blocks HER2 and HER3, and then the NRG1 fusion with its EGF-like domain cannot bind. Published in the New England Journal by Alison Schramm and colleagues, the NRG clinical trial showcasing an overall response rate of 30% with a duration of response of 11.1 months and a median progression-free survival of 6.8 months. And this clinical trial led to FDA approval for Zenocutuzumab in NRG1 fusion positive pancreatic and non-small cell lung cancer. But as you can see, there is activity across a broad spectrum of cancers, and it is interesting to see whether they're going to follow up with a tumor agnostic approval. Next, I would like to discuss MET amplifications. So c-MET and its ligand HGF, which can also bind to EGFR, are receptors that drive cancer progression through various different pro-survival pathways, such as the RAS pathway, the mTOR pathway, and the STAT pathway. And 3 to 5% of cancers display MET alterations. You have the classical MET exon 14 skipping mutations that we know from non-small cell lung cancer. We have point mutations in MET. Then what we want to discuss today are MET amplifications, and then there is obviously the MET fusion events. Presented at ESMO 2025, Zenocutuzumab adisutecan, or ABBV-400 TEMAP-A, is an ADC directed against MET and I'm going to show results from a phase 1 clinical trial. And this is interesting and important because MET amplification or overexpression is not automatically the driver of a disease, but commonly found. That is not to say that MET amplified cancers do not depend on MET signaling, but still a lot of cancers are MET amplified, and here this ADC can work. We see over 100 patients enrolled in this trial as of now, presented at ESMO 2025, 100 patients from non-small cell lung cancer, colorectal cancer, G junction cancers, and then other tumor types were enrolled in different cohorts plus expansion. And as you can see here, overall, the response rate was 46%, the highest response rates in non-small cells and GEA cancers, but also in biliary tract cancers. And the median duration of response was 10.9 months with a median PFS of 8.5 months and a median overall survival of almost 20 months in pre-treated patients. This is very encouraging data, and the numbers are not so small, so I'm really looking forward to see more patients being included in this clinical trial and then seeing if this holds true across a broader spectrum of cancers. Next, I would like to briefly discuss MTAP loss. Biologically, the idea that MTAP, which is lost in about 15% of cancers due to deletion of chromosome 9p21, leads to accumulation of MTA substrate, which then inhibits PRMT5. And if you then target PRMT5 selectively, which leads to synthetic lethality in MTAP-deleted tumor cells. This was shown in 2016 in this Cancer Cell manuscript. At the end of 2024, we've seen a tumor agnostic clinical trial of AMG 193, the first MTA cooperative PRMT5 inhibitor. This was published by Rodon and colleagues in Annals of Oncology, and you see a reasonable waterfall plot with responses across a broad spectrum of cancers. And at ESMO 2025, we saw a small phase 1 study. So this is clearly preliminary data. I'm showcasing this mainly because, as I said, 15% of cancers are MTAP deleted, and seeing other compounds like this MTA cooperative PRMT5 inhibitor entering the clinical trial arena is encouraging because it showcases that there is interest in following up on this targetable alteration. This is only 7 patients that are response evaluable. We have 43 overall response rate, 100% disease control rate, but caveat is obviously very, very small numbers here. But as you can see, the duration of response, it looks also encouraging. So I'm really looking forward to see expansion of this clinical trial with this compound. One of the most valuable tumor agnostic targets is HER2. You're all familiar with Destiny Pan-Tumor 02, the pivotal trial by Fundameric Bernstam and colleagues that led to the tumor agnostic approval of trastuzumab deruxtecan for HER2 3+ cancers by the FDA. And this compound has also been submitted to the authorities in Europe for approval in HER2 3+ cancers. And at ESMO 2025, we saw the final data overall response rate in HER2 3+ cancers 52%, a PFS of 11.9 months in this population, and an OS of 21.1 months in IHC 3+ cancers. I see that my face is in front of the last line. So what this says is even in pre-treated patients, so patients that had received HER2-directed therapy, the overall response rate was 36.8%. So clearly trastuzumab deruxtecan is an ADC, and we've also seen zongertinib. This is a HER2 selective tyrosine kinase inhibitor being published last year in JCO, and here you see that lung cancer patients, and here we have an approval already, respond well to this drug, but also the non-lung patients. And Boehringer Ingelheim is following up with tumor broad basket trials, and I'm excited to see whether this is going to lead to a tumor agnostic approval, or at least broader application of this agent. Now moving into targeting KRAS or RAS, and here I would like to concentrate on innovative strategies, not so much in the drugs that have been discussed quite broadly in pancreatic cancer, but to set the perspective here a little, over 4 million patients will be diagnosed with KRAS mutant cancers worldwide each year. Obviously, very, very frequent in pancreatic cancer, where you reach up to 90% of patients that are RAS mutant. And importantly, while maybe a decade ago people would have argued that targeting RAS is impossible, we now see a variety of KRAS and RAS inhibitors being developed in clinical trials. We all know about sotorasib and adagrasib that are approved for G12C mutant cancers, not in a tumor agnostic fashion, but in a disease specific fashion. And these mutant specific inhibitors target a single KRAS allele. They are very precise, but what we've now seen is that resistance mechanisms are frequently emergent, and obviously they're allele restricted. If you then move from mutant-specific inhibitors to pan-KRAS inhibitors, you block multiple KRAS alleles. The different drugs that are now in clinical trials, and I just give 2 examples here, they have a broader coverage, but what we've seen in the early clinical trials is the toxicity expand, because you're also hitting KRAS, wild-type KRAS in some cases. Moving even broader, pan-RAS inhibitors that then target all RAS isoforms, not only KRAS, but also N and HRAS. You have tri-complex on-state inhibitors like RMC-6236 or RMC-7977. You have maximal coverage, but a high risk of a wild-type toxicity. So this is really going to be a mix and match when you go in with these broader and very extensive inhibitors. And then, and this is what I would like to focus on a little bit today, are alternative strategies like PROTAC, so degrader proteins, small RNA interference, or targeting other upstream mechanisms. And here the obvious benefit is that you bypass resistance mechanisms. And what I foresee is that these different strategies are going to be combined to prevent or overcome resistance while trying to minimize toxicity. I've chosen a couple of approaches that I found interesting in 2025. The first one coming from Weinberg and colleagues, being published in Nature Medicine 2025, where the colleagues tested a mutant KRAS specific vaccine targeting lymph nodes. in pancreatic cancer and colorectal cancers. And those were patients with a very high risk of relapse. And they were then treated with this ELI 022P and assessed by imaging, immunogenicity, and ctDNA follow-up. Obviously, this is very preliminary data, small numbers, but what you can see is if there is a immune response as determined by a T-cell response, you can see that the median overall survival looks promising compared to those patients that do not elect a T-cell response. And the same is true for the median relapse-free survival. Those patients, and I said, those were patients at very high risk for relapse, relapsed within 3 months if they did not have a response with regards to their T cells, but the median RFS was not reached when a certain threshold was reached. Secondly, showing some data from ESMO 2025, the first in human phase 1 study of a T cell receptor T therapy targeting KRAS G12 in metastatic solid tumors. And here you see the difference between the classical CAR T cells that look at antigens that are expressed on the cell surface, and you build your CAR against it, and TCR engineered T cells, TCR-T, recognize intracellular tumor-derived antigens that are presented by MHC molecules and can thus access a much broader antigen repertoire than CAR T cells. So we're looking at TCR-Ts against KRAS G12V. In this phase 1 study, 14 patients were included, almost all of them having stage 4 disease and a median of 3 previous treatment levels, different dose levels. And if you look at the overall response rate in those with dose level 2 or above, those were 10 patients, 50% had a measurable response, which included 5 partial responses and a disease control rate of 100%. Again, very small numbers, but promising with regards to a new treatment option. And looking at the median PFS in those with dose level 2 or above, 5.8 months in patients with more than 3 lines of therapy is encouraging, with the caveat again and again that those are small numbers. So coming to an end, what was practice changing in 2025? This is difficult with regards to tumor agnostic treatments because we haven't seen novel tumor agnostic approvals. But what I believe becomes clearer and clearer each year is that comprehensive molecular tumor profiling keeps expanding due to a growing number of molecularly guided treatment options. So more and more patients need broad testing. I would argue that targeting HER2 is now broadly established as a tumor agnostic target, and further therapeutic options are entering the clinic beyond just trastuzumab deruxtecan. I've shown you the data on zongertinib. Emerging tumor agnostic targets include KRAS and RAS inhibitors. I believe this is going to be very, very important in the future. And I've shown you interesting data on targeting MET amplification using ATCs. I've shown you PRMT5 inhibitors and Zenocutuzumab targeting NRG1. Coming in 2026, I believe this is going to be the year for the pivotal clinical trials and hopefully approvals when it comes to targeting KRAS. And with this, I would like to extend my gratitude to Gil Morgan and the OncoAlert family for letting me be part of this OncoAlert Colloquium 2026. And I'm happy to discuss this talk in the chat.